Assalamualaikum and hello everyone. This is the video report for STB1083 Biochemistry Practical 3 by Subgroup 5. Before we dive into the experiment, I'm gonna introduce our group members.
Hi, my name is Nuralia Adila binti Rudirukimin with a matrix number of 97658.
Hi everyone, my name is Nurintisna Batusha binti Ahmad Raf. My matric number is 97736.
Hi everyone, my name is Shasha Mera Alia binti Muhammad Farah Asri with matrix number 97811.
Hello everyone, my name is Nushahira binti Hafid with the matrix number of 97739.
The title for this practical is Physical Properties of Different Lipids and Fatty Acids. The objective of this experiment is to identify lipid-containing products in everyday food and materials and prepare them for analysis, to observe and record the appearance and odor of different lipids and fatty acids, to compare the solubility of lipid in the polar solvent water versus the non-polar solvent toluene, and to relate these physical properties of lipids to their classification as a distinct group of biomolecules. There are 6 materials for this experiment, which are microcentrifuge tube, spatula, dropper, distilled water, toluene, and 3 selected lipid-based food, which are olive oil, shortening, and ghee. Next, we are gonna dive into the procedure. First, 2 sets of 3 separate microcentrifuge tubes were prepared. 5 drops or a small amount of solid, about 3 to 4 granules, of all the lipid-containing product was added. So next, their appearance and smell was described in the table above. For ghee, it formed thick cream yellowish color. For shortening, it formed heterogeneous layered thick cream white color. And for olive oil, its appearance is liquid yellowish color. For ghee, it produces slightly buttery-like aroma. For shortening, it produces unpleasant smell, and for olive oil, it produces fruity scent. For set 1, we are gonna test the solubility of lipids in polar solvent. About 1 milliliter of water was added to each centrifuge tube. The tubes were capped and shaken to mix. The observations were recorded in the table. For set 2, we are gonna test the solubility of lipids in a non-polar solvent. 0.5 milliliter of toluene was added to each sample. The tubes were capped and shaken to mix. The observations were recorded in the table. For conclusion, in this experiment, we examined the physical properties of lipids and fatty acids, such as appearance, odor, and solubility, and also confirmed their non-polar nature. Olive oil, shortening, and ghee were soluble in toluene but insoluble in water, reflecting their hydrophobic character. This finding aligned with the theory that lipid solubility is influenced by non-polar hydrocarbon chain. The experiment also highlighted how lipid structure relates to their role in energy storage, membrane formation, and biological functions. Overall, it deepened our understanding of lipid properties in biochemistry. That is all from us. Thank you.